Now I will explain you about the modern analytical techniques in mass spectrometry and the molecular techniques in food analysis and testing. The demand for the quality and the safety in the food products have made analytical techniques critical in the food analysis. Among these, mass spectrometry and the molecular techniques have emerged as vital tools to ensure food authenticity, detect contaminants and assess nutritional value. This explores these techniques focusing on their principles, applications and importance in the food industry. Now coming to the mass spectrometry in food analysis. It's a powerful analytical techniques that identify and quantifies compounds by measuring the mass to charge ratio of ionized molecules. It is widely used due to its precision, sensitivity and ability to analyze the complex mixtures. Mass to charge ratio is to describe the charged particles such as ions. It is the ratio of the mass of the particle to its charge. For example, if an ion has a mass of 12 AMU and a charge of plus 2, means 2 electrons, its m by q, that is mass to charge ratio is equals to 12 AMU by 2 electrons. Then we get 6 AMU by electrons. This is the mass to charge ratio. Now let us discuss about the components of the mass spectrometer and its applications in food analysis. First, coming to the inlet. This is the entry point where the sample is introduced into the mass spectrometer. Whatever the food sample we wanted to test, we need to introduce here. The sample can be solid, liquid or gas form depending on the type of analysis. In which method the sample will be introduced? There are different types, that is direct insertion. Direct insertion means a solid or liquid sample is inserted directly into the ionization source. And the next method is the gas chromatography interface. It uses the volatile compounds that are first separated in the gas chromatograph. Next method is the liquid chromatography interface, used for the liquid samples or the mixtures that are separated via the liquid chromatography. Now coming to the source, that is ionization. The ionization source converts molecules into gas phase ions, which are necessary for mass analysis. There are different types of ionization methods used here. One is electron ionization, means a high energy electron beam bombarded the sample, removing an electron to produce the positive ions. Next method used here is electrospray ionization, that is ESI. A liquid sample is passed through a fine capillary, creating charged droplets that evaporate, leaving gas phase ions. Coming to the analyzer. The analyzer sorts ions based on their mass to charge ratio, that is m by z. And the types of analyzers used are quadrupole. It uses the oscillating electrical fields to selectively transmit ions with specific mass to charge ratio. Common in routine analysis due to its accuracy and speed. Next type of analyzer used here is time of flight, TOF. Measures the time ions takes to travel a fixed distance. The lighter ions reach the detector faster, providing high resolution results. Next type of analyzer is magnetic sector. Uses a magnetic field to bend ions trajectories. Ions with different m by z values follow different paths. Next analyzer is ion trap. Traps ions using the electrical fields and sequentially ejects them for detection. It is useful for tandem mass spectrometry. Next one is the ion detector. It converts the incoming ion signals into an electrical signals that can be measured. And the types of detectors used are the electron multiplier, which amplifies ion signal by causing secondary electron emissions. Faraday's cup measures ion current directly, less sensitive but robust. And next ion detector is microchannel plate. This is highly sensitive, used in TOF systems. TOF system means time of flight analyzer systems. Next is the data system, which collects and processes the electrical signals from the detector, produces a mass spectrum which is a graph of ion intensity versus m by z. Its function is to determine the molecular weight and structural information of compounds and provides quantitative data, which is the concentration of ions, and enables quantitative analysis, that is identifying the unknown compounds. Next, coming to the applications in food analysis. Detection of contaminants and adulterants. This mass spectrometry detects pesticide residues, heavy metals and the mycotoxins in food. Example, liquid chromatography MS. Liquid chromatography mass spectrometry is used to identify the pesticide residues in the fruits and vegetables. Next one is the authentication of the food products. It ensures the geographical origin and authenticity of the products like honey, olive oil and the wine. Next one is the analysis of nutritional content. It quantifies vitamins, amino acids and the fatty acids. Next one, mass spectroscopy is used in the detection of the food frauds. It identifies the mislabeling or the substitution of the ingredients. For example, identifying the horse meat in processed beef using the mass spectrometry. Coming to the advantages of mass spectrometry, it is highly sensitive and accuracy and able to analyze small quantities of the samples and versatility in detecting a wide range of compounds. Coming to the limitations, it is highly cost and requires skilled operators for data interpretation. Coming to the molecular techniques in food analysis, it is essential for detecting the foodborne pathogens, identifying the genetically modified organisms and verifying the food authenticity. And these methods rely on the analysis of the DNA, RNA or proteins. Prominent molecular techniques used are polymerase chain reaction, that is PCR, which amplifies specific DNA sequence to detect pathogens or the genetically modified organisms. Example, identifying Salmonella or the E. coli in the raw meat. Next is the quantitative PCR. It provides quantitative data on the concentration of the DNA targets. It is useful for detecting allergens in the food products. I have already explained this all in the before classes. Please check it out once. Next, coming to the next generation sequencing. It offers comprehensive genomic analysis and applications are microbiome analysis in fermented foods and detecting unknown pathogens. Enzyme linked immunosorbent assay ELISA. It detects proteins or allergens using antigen antibody interactions, commonly used to detect gluten or the peanut allergens. Biosensors. It detects specific molecules like toxins or pathogens. An example is rapid detection of aflatoxins in the cereals. Coming to the applications of molecular techniques, food safety, detection of pathogens such as Listeria and Campylobacter and the noroviruses. And it is also applicable in food authenticity, identification of animal or plant species in the processed foods. GMO analysis, differentiating genetically modified crops from the non-genetically modified counterparts. And self-life assessment, monitoring the microbial spoilage over time. Coming to the advantages of the molecular techniques, it is highly specific and sensitive, rapid and reliable results and minimum sample preparation is used for the molecular techniques. Coming to the challenges, high initial investment is needed and a complexity in interpreting results for mixed food matrices. And the applications of mass spectrometry and the molecular techniques has revolutionized food analysis and testing. Their ability to produce accurate, sensitive and the rapid results ensures food safety, authenticity and nutritional quality. However, the challenges of the cost and the complexity must be addressed to make these technologies more accessible. With continued advancements, these techniques will remain pivotal in meeting global food safety standards and the consumers' expectations.